Today we're going to be briefly discussing the countercurrent exchanger, but mostly we're going to be discussing the countercurrent multiplier. So we start off drawing another kidney. I'm drawing it sideways this time so that we can kind of follow the course of the angles of the renal pyramid. So I'm drawing the renal pyramids in here, and I'll go ahead and fill in the renal artery and the renal vein and the ureter. This time we're going to go ahead and fill in some numbers. We're going to fill in the cortex 300 milliosmoles, MOSM. 300 milliosmoles is the osmolality, or basically the salt versus water content of pretty much the entire body, all of the fluids of the body, all the blood, all of the interstitial fluid, all of the lymph, aqueous humor in your eyes, all the fluids of your body. Except deep inside of the medulla, it starts to become more salty than that, and so the numbers go up. 400, 700, 900, all the way down to 1200 deep inside of the medulla, kind of at the base of those, or at the tips of those renal pyramids. The number is going to be 1200, quite salty inside of the kidneys, inside of the medulla of the kidneys. So let's go ahead and draw one single renal pyramid so we can explore exactly what's going on inside of there. So these are called renal pyramids or medullary pyramids. And as long as you're just, above or outside of the pyramid in the cortex, it's going to be 300. But very quickly, as you move deeper inside of the medulla or inside of that pyramid, you're going to have numbers that increase. Again, 300, 700, 900, all the way down to 1200. So what we're saying is that deep inside it is quite salty, and there's this gradient that is established. And you have to understand that this is quite tricky, and this is a point that's going to come up again, but I don't know if I'm really going to emphasize it again, so I want to emphasize it now. It's quite difficult for the body to maintain any kind of gradients. The body dislikes, equilibrium is what we go for, actually. We always want things to be equal, and so there's a continuous fight against the body's strive for equilibrium. Water is always going to be seeping into the deep medulla in order to break up all that salt content. The salt is always going to be diffusing out into the cortex. And so we always have to reestablish this salt gradient, and that's actually the point of the countercurrent multiplier. And the importance of it will be coming up as we continue this lecture. But just for now, just understand that it's important to have that salt gradient there. Okay, so let's draw a collecting duct. It's that stripe that makes its way through the medullary pyramid, through the renal pyramid. And for context, I'm also going to draw a nephron here. I'm going to draw the glomerulus. I'm going to draw a little glomerular capsule, the PCT, the nephron loop, which is just kind of a hypothetical length, and then the DCT, and the DCT empties into that collecting duct. And remember, there will be multiple nephrons, emptying into this collecting duct. And also it should be said that there's actually two different types of nephron loops. There's one that's closer to the cortex called a cortical, and there's one that goes quite deep called juxtamedullary. We don't really parse the difference between them in this class, but just be aware that they exist. So let's take a look at that nephron loop. First of all, we go ahead and draw the vasa recta. The vasa recta is the peritubular capillary that surrounds the nephron loop. And the reason the vasa recta is there is to bring oxygen, nutrients to the cells, which are actually quite metabolic here in the walls of the vasa recta. The vasa recta is called the countercurrent exchanger because as this blood supply, it's very necessary for the metabolism of these cells in the nephron loop, comes along, we don't want it messing around with that gradient. So it has two characteristics, the vasa recta does. It has a slow blood flow, and also the vessel walls of this particular capillary, the vasa recta, are very leaky. Therefore, we have free exchange of both sodium and water so that it doesn't mess around with that gradient that we're trying to establish. So the vasa recta is called the countercurrent exchanger because it helps to maintain that osmotic gradient. Doesn't mess around with it. It is a fluid that comes in, dips into the deep medulla, and dips right back out without ever causing the problems that could ostensibly be caused if you had, an irresponsible fluid coming into the region. It's very likely that it would start to mess around with that gradient. Irresponsible fluid. So this is the countercurrent exchanger. It allows exchange of fluids in and out and just reestablishes that gradient as it dips in and out. Okay, it's important to discern between the cortex and the medulla. The cortex, again, is just above or outside of that medulla and of the medullary pyramid. And of course, as soon as you have a medullary pyramid, you are inside of the medulla. So there is a distinct line that separates the cortex and the medulla, but it may be important to say that there's not actually a membrane there. It's just there's not a membrane, for example, like keeping the salt gradient in and out. But anyway, the cortex is just outside of the pyramids, and the medulla is as soon as you dip into those pyramids. All right, let's go ahead and draw glomerulus, glomerular capsule. We're just going to draw the blown up version of the whole thing, the whole nephron, with a couple of variables here. And I am skipping a lot of the labeling, for better or for worse. We're skipping a lot of the labeling here. So, glomerulus, glomerular capsule, PCT, and now we dip into the nephron loop. And as we do this, we keep everything nice and skinny to differentiate the skinny part from the fatter part. Now this structure of the nephron loop, we're going to be calling the descending limb of the nephron loop. And this part here, although I didn't label it, this is going to be the ascending limb of the nephron loop. Remember, that's one direction only for filtrate to move on through all of this stuff. And so, yeah, it goes down through one end and comes up through the other every time. And here's our DCT. And what we're going to do is we're going to go ahead and draw two different collecting ducts because we want to run two different scenarios. And so we're going to basically draw a little interrupt line there, and we're going to draw, here's the DCT emptying in this one on the right-hand side. A little sloppy there, sorry. And then there's another collecting duct. Let's say that the DCT instead empties into this guy. So you understand there's two different possibilities here. This DCT, everything is the same up until this point, and then there's two different scenarios. We're going to go ahead and draw a dotted line, and we're going to be differentiating between the cortex and the medulla. And just a remind that the glomerulus, glomerular capsule, the PCT and the DCT are all in the cortex, and then the nephron loop dips down into the medulla. And additionally, we're going to go ahead and draw the one number we know very well, which is that there's 300 milliosmoles throughout the entirety of the cortex. So we're going to put that everywhere to remind ourselves that everywhere in the cortex, the saltiness of the cortex, the osmolality is going to be 300, which is the same as, the synovial fluids inside of the elbow and your lymph. And all of the fluids of your body are 300, and so is this fluid in the cortex. Except, of course, when you dip down into that medulla, as soon as you dip down into the medulla, it's at 400, and then all the way down at the elbow of the nephron loop, it's 1200 milliosmoles, and then we'll fill in some other numbers in there to establish a gradient between the 400 and the 1200. Let's go ahead and extend those collecting ducts a little bit further. And you may recall that we use the color orange to establish filtrate, and so and also urine. And so we're going to say at the end of these collecting ducts, just to remind you, once that collecting duct moves all the way through the renal pyramid, it's going to move, through that renal papilla and into the minor calyx. And at that point, the fluid is going to be urine. Now we're looking at two different scenarios here. In one of the scenarios, we have ADH, antidiuretic hormone, which was released, you may recall, from the posterior pituitary lobe. And the role of ADH is going to be to put these little proteins into the walls. It's a little tunnel going through from one side of basically from the lumen of the collecting duct into the interstitial fluid next to it. And it turns out these are aquaporins, water pore proteins. They are proteins that allow water to flow through. Now let's take a little bit of time to understand in which direction water will be flowing through these aquaporins. In order to do this, let's go ahead and establish what's going on with the filtrate. what is the condition of the filtrate as it enters into the collecting duct? Well, what we can say is that the condition of the filtrate is that it is quite dilute, actually less dilute than most fluids of the body. Remember, most fluids of the body are at 300. Well, it turns out the fluid in the DCT is about 100, and we'll talk about exactly why this is in a moment. But and by the way, it's simplifying the story a bit, because there are certain scenarios where the fluid actually will not be quite this dilute. But I think it's useful to make the DCT fluid, the filtrate moving through the DCT at 100, and empty it into the collecting duct. And so you look into both scenarios here, a situation where you have ADH, and you have a situation where you don't have ADH. And if we have this filtrate entering into both situations at 100, it's useful to be able to compare exactly how it changes as it moves through these two different tubes. Well, as a reminder, remember that we're moving through the deep part of the medullary pyramid, and therefore we can go ahead and write our numbers in. We can write numbers grading from 400 to 1200 to indicate that it's getting saltier and saltier. So it turns out that if you have aquaporins inserted into the walls of the collecting duct, that water is going to leave the filtrate and re-enter the body. This is tubular reabsorption right here. We are taking stuff from inside of the filtrate, and we're putting it back into the surrounding interstitial fluid, which is where it is able to re-enter the body. And so what's going to happen is that as we move into this saltier and saltier atmosphere, as the filtrate moves through this salty pyramid, this renal pyramid, what's going to happen is that it's going to lose so much water that it itself is going to become quite salty. And so what we're going to do is we're going to write numbers that sort of lag behind the surrounding interstitial fluid, because it's not going to be a perfect osmotic experience. You're not going to lose so much water immediately that you're going to have equality between the saltiness of the filtrate and the saltiness of the surrounding interstitial fluid. That would be considered isoosmotic. But that is not actually what we have. We have a bit of a lag here, because we're not releasing all possible amounts of water. We're releasing some water, and so we get a filtrate that is saltier and saltier as it moves through the collecting duct, but it never really achieves 1200, unless you were incredibly dehydrated, like 3 days without water kind of a thing. That would be then you might actually achieve that level of saltiness, but under normal circumstances you would not be achieving that level. So what we end up with then is urine that is salty. It's going to be dark in appearance. A person under these conditions probably would not have to urinate very often. And this urine would have a high specific gravity. Well, let's look at the other condition. If there is no ADH present and there were no aquaporins inserted, then what's going to happen is the filtrate is actually going to remain quite dilute at 100 or so. As it moves through the collecting duct, it doesn't really matter the saltiness of the surrounding interstitial fluid. There is no way for water to leave, and so you end up with your urine being quite dilute. This is going to be a low specific gravity, close to 1. So water is a specific gravity of 1, and then anything a little saltier than that would go up from there. So anything very, very dilute would be close to 1. So a low specific gravity here, and this person would probably have to urinate quite often. Well, let's talk about the different conditions. What's the practical side of this? Well, it turns out if a person is dehydrated, it is likely that they will produce a lot of ADH, have lots of aquaporins, and develop that ability to reabsorb water back into the interstitial fluid, and therefore have higher specific gravity urine, saltier urine. But a person who is fully hydrated would no longer really need to reabsorb water back into the body, and therefore they would have the ability just to go ahead and get rid of it. They would not need ADH, they would not need aquaporins. Reabsorbing water from the filtrate would not be an issue, and boy, it took me a long time to write this word. A person who's hydrated, basically, is in this situation. But there's also two other possibilities here. If you simply do not produce enough ADH, then that also would lead to this issue even if you were dehydrated. So for example, a person who drinks alcohol famously blocks the production of ADH, and therefore a person who has been drinking alcohol will tend to urinate quite a lot even if they are not fully hydrated. It's a good idea to drink water, lots of water, if you are drinking lots of alcohol in order to keep your body hydrated, because otherwise it's going to act like it is hydrated even if it's not, and so you're going to go ahead and get rid of lots of fluids even if you actually need to be retaining them. So alcohol blocks ADH, and then there's another condition called diabetes insipidus. Diabetes insipidus. Now, diabetes insipidus, the term diabetes means excessive urination. And there's a number of conditions that cause excessive urination. The one that we usually think of is diabetes mellitus, where we have the inability to properly control our blood glucose, oftentimes because insulin is in low production. But this is different. Diabetes insipidus is caused by ADH not being properly produced by the posterior pituitary lobe for whatever reason. There's a number of different conditions that can cause this issue as well. And so in this case, a person will tend to be very thirsty and drink a lot of water, and they'll also urinate quite a lot. So they do have diabetes. They urinate a lot, diabetes, but this specific type of diabetes is called diabetes insipidus. So both of these scenarios, just kind of, practical ways that you can still not produce enough ADH, and therefore your urine remains very dilute and your filtrate remains very dilute, even if you're in a condition where you should be retaining water. You probably won't be if you don't produce ADH. You won't produce aquaporins, and you won't reabsorb water. Let's go ahead and move on to discussing how we got to that very dilute filtrate in the first place. And so we're going to fill in some numbers, kind of zeroing in a little bit because we had to get really small with these numbers. So what happens is we have filtrate. If we just observe the numbers of the filtrate is going to be at about 300 as it enters into the nephron loop. But then as soon as it gets into that descending limb of the nephron loop, it's going to become saltier and saltier. And we'll talk about how. At the very elbow, the base of the nephron loop, it's going to be at about 1200. And then as it climbs out of the ascending limb of the nephron loop, it is going to get more and more dilute. So the question is how? Let's go ahead and draw some red arrows. We're going to be indicating that there's going to be sodium ions that are going to be pumped out of this thick segment of the ascending limb of the nephron loop. So in the ascending limb of the nephron loop, we're going to be pumping out sodium ions. And I like to establish this right away because this is actually pretty much what causes that saltiness in the deep medulla. That pumping out of the sodium ions out of a filtrate that down at the very lowest part of the nephron loop was very salty at 1200. You'll notice that the number is very precipitously. As the filtrate comes out, the numbers continue to get lower and lower until they're at 100, which is quite dilute. So we're going to go ahead and write the 5 steps of the countercurrent multiplier system, and you'll notice the different colors here. I already mentioned step 3, quite important, the pumping out of these nephron loop, the pumping out of salt out of the thick segment of the ascending limb of the nephron loop. But step 2 is the thin segment. And what happens in the thin segment, just that small part, the first part of the ascending limb, is that some of the sodium ions simply diffuse out as the filtrate goes into a more and more dilute. As it rises up through the medulla, its surrounding interstitial fluid becomes less and less salty, so there's some amount of diffusion happening as this very salty filtrate begins to progress through that region. Well, since we've been working backwards, here's our very first step. The last thing we're really discussing is the water leaving out of the descending limb of the nephron loop. So it turns out the descending limb of the nephron loop is permeable to water, but not salt. And the ascending limb of the nephron loop is permeable to salt, but not water. So you see the difference there. In the descending limb, water is able to simply move out through osmosis as the filtrate, which again started off at about 300, the same as, all fluids of the body, the same as the blood plasma. It started off as 300, but it quickly loses water as it moves into this saltier and saltier atmosphere until finally, in the very lowest part of the nephron loop, there we have it. We've lost so much water that we have established a filtrate that is quite salty at 1200. This gives us the ability then. It's tricky thinking about this. You have to think about it several times. You have to go through this story several times to keep until it clicks. But basically you have this very salty filtrate because it has lost so much water that now you have the ability, as it climbs out of the nephron loop in the ascending limb, that now has the ability to lose all of that salt. And the salt is diffused out in the thin segment and then pumped out in the thick segment of the ascending limb, and that in turn, all that salt coming out of there is what allows for that increasingly salty, increasingly high osmolarity inside of the deep medulla. Well, after all that work with the salt and the water, it seems almost anticlimactic to discuss anything else, but we do have to mention urea. Urea is going to be also diffusing, oftentimes directly from those collecting ducts. Is going to be entering into that interstitial fluid. This also is a substance that helps to establish a high solute atmosphere in that deep medulla. And so it doesn't actually matter whether it is a situation where ADH is present or not, or aquaporins are present or not. Urea will be leaving out of those collecting ducts and re-entering the interstitial fluid and allowing for more substances, which just increases the osmolality of the deep medulla. And then lastly, as we said earlier, and we spent some time on this one, so I will say that the fifth step is water retention, water reabsorption. And this is, it's kind of really the point of the whole thing. The point of having the countercurrent multiplier is to have a salty medulla so that you have a choice. If you are dehydrated, you're going to produce some ADH, you're going to make some aquaporins, you're going to reabsorb water from the filtrate as it moves through the collecting ducts. And that's step 5. But notice that step 5 would never happen if you didn't have a nice salty atmosphere, a nice salty interstitial fluid surrounding that collecting duct in the first place. So you need salt. Steps 2 and 3, you need salt. But of course, step 5 doesn't even really matter if you are fully hydrated, or if your body doesn't create enough ADH for some reason. Then what's going to happen is that filtrate is not going to leave out of the collecting ducts, and therefore it's going to remain inside of the filtrate. Well, I want you to keep in mind 3 terms that I want you to think about. If you can understand how to put these terms into context in terms of this lesson, this will be a good sign that you really understand everything. The terms are isoosmotic, which means same osmolality, same saltiness. Hypoosmotic, which means less than, a substance with less saltiness. And hyperosmotic, which is a term that means more saltiness. So let's give a couple examples of this. Let us compare the fluid inside of the DCT with the surrounding interstitial fluid. Remember, the fluid inside of the DCT, at least according to the way that I teach this lesson, is always going to be 100, which is pretty dilute, and the surrounding interstitial fluid is going to be 300 milliosmoles. Well, then we would say, well, the filtrate inside of the lumen of the DCT is hypoosmotic to its surrounding interstitial fluid. Basically, it is less salty than the surrounding IF. Let's look at one more example. Let's take a look at the filtrate in the very deep level of the nephron loop. Remember that the filtrate in the deep level of the nephron loop is 1200, and the surrounding interstitial fluid is also 1200. So this would be an example of isoosmolality, or an isoosmotic situation. So we would say the filtrate in the deepest part of the nephron loop is isoosmotic to its surrounding interstitial fluid. So give yourself a few examples like that, see how you do. Think about the fluids inside of the collecting duct in the presence of ADH versus not in the presence of ADH. You could also use terms like the aquaporins. You could think about the PCT and its surrounding interstitial fluid. You could think about the descending limb of the nephron loop, the ascending limb, both the thin segment and the thick segment. just think about running some of those scenarios. Again, if you can establish, if you can understand how to put these statements together, then you'll be doing pretty well, and you'll understand the lesson as a whole pretty darn well.